Hello, everyone. We're going to have 3 lectures on urinary system this week. Okay, 3 lectures. So I will send you an email, give you instructions. For the first lecture, we're going to cover urinary anatomy, and then the second one will be urinary physiology. The third one will be acid-base balance, regulation of acid-base. Okay, so for the first lecture, we're going to focus on the urinary anatomy, and you need to read your book from page 974 to uh, page 986, 974 to 986 first. That's the first step. And second one is I post a assignment on Canvas. It's called Unit 4 Homework Urinary Anatomy. The second part, after you finish reading, you should work on that homework. Okay, get the homework done. And then the third step is to watch this video. Okay, so please do that. Do this in this sequence. All right, read, do homework, and then listen to this lecture. All right, so let me have the share screen here. Okay, we're going to start urinary anatomy. The notes are posted on Canvas. Let me see. Here we go. So urinary system, if you think about this, can you come up with the major organs involved in this system? Kidneys, yes. Anything else? Urinary bladder, yes. Anything else? Ureters, yes. Okay, ureters is the tubular structure carry the urine from kidneys to the urinary bladder. And then the last part will be the urethra. Good. So kidneys are in the abdominal cavity, okay? And the left and right side is not at the same level. It's uneven, but it's not showing here. Okay. That is okay. So from this posterior view, you can see the last pair of ribs are the bony protections for kidneys. Okay, and if you look make a cross section and look from superior view, you can see this is anterior, posterior. This is where it make the cut. Okay, and then you can see the protection layers on the surface of a kidney. So here's the kidney right here, okay? So on the surface of a kidney, we have something that is the connective tissue is called renal capsule on the surface of kidney protection layer. And then superficial to that is adipose tissue, okay, adipose tissue for cushioning, okay? And then outside of adipose tissue, there's another connective tissue layer, okay? It's called a renal fascia. So basically, that's the 3 layers on the surface of kidney to protect kidney, okay? Of course, on the surface of that, we have the bony protections. Okay, so major artery and veins, the blood vessels in and out of kidneys, renal artery, the arteries, renal arteries are branched out from aorta, okay, and renal veins drain blood to inferior vena cava. All right. So these are the major organs inside in the urinary system. In terms of function, what is the main function for urinary system? Do we know? To filter blood. Yes, to filter blood. In terms of the processes, the mechanisms for urinary system to filter blood, we have four big parts. First part is called filtration. Okay, blood will be filtered in urinary system, actually in the kidneys, right? Filtration. The second part is called reabsorption. So most of the filtered materials will be reabsorbed back into blood. This process is called reabsorption. The third part is called secretion. When we talk about the secretions, we're talking about in this urinary system, there are mechanisms we can use to actively transport certain materials out from blood into the filtrate and then into the urine. For example, hydrogen ions, to balance the acid concentration, we can actively use mechanisms to actively remove hydrogen ions from blood into the filtrate and then into the urine. So that's the third part is secretion. The fourth part, sometimes people will list this part, as one step. That's called excretion. Excretion is just move materials in the urine from urinary system to outside. That's excretion. Okay, so these are the major steps or major mechanisms involved in urinary function. So today we're just going to focus on the anatomy part. Next time we will talk about the physiology part. Okay, so if we look at the kidney structure, okay, so from this picture you can see the outside here, the lighter color part is called the cortex, okay. Deeper part is called medulla, okay, medulla. And you can see the cortex structure, this lighter part can extend it deeper into medulla area. This part, okay, it is still cortex structure, extended deeper into medulla. And then medulla structure, inside the medulla you can see this very nice pattern structure is called a renal pyramid. Okay, so there's a compartment in the medulla is called a renal pyramid. So if we enlarge this renal pyramid and we take a look, see here, okay, outside layer right here is a cortex, and then here this part will be the renal pyramid. Okay, so if you see there are many like a parallel looking tubular structures in the pyramid, that's right here. You see kind of look like a parallel fibers, but it's really the parallel tubular structures. I'll give you the names of them. Okay, but if you look at the cortex here, we do have these tubular structures, this yellow part, but they're not so parallel to each other. They're more like a folded, we call it convoluted tubules in the cortex. But in the pyramid, you see mostly parallel tubules. Okay, all right. And then, the blood will be filtered in the kidney, and then the filtered fluid is called a filtrate. Okay, so this filtrate is going to travel through this kidney structure from cortex to medulla and toward to the end here. This is the ureter. Okay, so in this traveling path, majority of the materials will be reabsorbed back to blood, And then finally, the leftover fluid in this area is called renal pelvis. That is the fluid we call urine. Okay, so before this point, in the process, okay, of making urine, the fluid is called filtrate. Okay, all right. So the main function for kidneys is to filter blood. All right, because of that, we need to know how does the blood travel through the kidneys and how can the blood being filtered. Okay, so if you think about filtration, any kind of materials exchange between blood vessels and surrounding environments has to take place in around the capillaries. We already know that, right? Only the capillaries, the wall is thin enough to allow material or gas exchange take place. Okay, so let's take a look at this blood trace. Okay, blood running through the kidneys. All right, start from aorta, we already know, and then we get to the renal arteries. Okay, we know the in between, right? Aortic arch, and then what? Thoracic aorta, abdominal aorta, and then we get to the renal artery. Okay, and then from renal artery, this is renal artery, branch out to form segmental arteries. Okay, once the renal artery branch, it will form the segmental arteries. Okay, and then the segmental arteries will travel and then start to branch out again. Then this one is called a lobar artery. Lobar artery. Let me see if your book is using. Okay, actually, your book took out this lobar artery. Okay, so we're going to follow your book. So you can cross out this part. Let me see if I have a. I can't. Can I write? Oh, here. Okay. Yes, very good. Okay, so we're taking out this lobar artery, huh? All right. So it's the aorta branch to form renal arteries. Renal arteries branch to form segmental artery right here, and segmental arteries branch to form interlobar arteries. These interlobar arteries are between the pyramids. If you see, these arteries between the renal pyramid is called interlobar artery. Okay, and then this interlobar artery will branch to form these arcuate arteries. See, these arcuate arteries are the arteries between the cortex and the medulla. Okay, right at the border. Arcuate artery between cortex and medulla. And then from arcuate artery, these are at the like arch looking arteries, branch out many branches into the cortex structure. These are called interlobular artery. It's not an interlobar, okay? It's called interlobular artery. Let me see your book. It's also called cortical radiate artery. So you can write it down here. I don't know if I can't write down. I can't write anything. Let me see. No, I cannot write. No, I cannot write. So you can do that, okay? So this interlobular artery is also called cortical radiate artery. That's these little arteries branched into the cortex, okay? And then from the cortical radiate artery, it's going to form the smallest artery. It's called afferent arteriole. Look, arteriole is the smallest artery. Okay, afferent is blood going in somewhere. It's afferent arterial. And then blood coming out is efferent arterial. In between, there's the capillaries. All right, this picture is not big enough to show that, but just think about, okay, between these two smallest arteries, there's a capillary bed in between, okay? So from the efferent arterioles, and it's going to connect it to another capillary bed, okay? It is called peritubular capillaries and visa recta. I will explain this in a minute, okay? And from these capillaries is going to connect it to the interlobular vein or the cortical radiate vein, the same thing. And then go to the arcuate vein. So that's arcuate vein right here. In cortical radiate veins, these blue ones over here in the cortex, arcuate vein. And arcuate vein merge to become interlobar veins right here in between the pyramid. And then from interlobar veins, going to the renal vein here, merge to the renal vein. On the venial side here, there's no segmental vein. All right, so make sure you remember that. Only there are segmental arteries, but there's no segmental veins. Okay, so now if you look at this trace here, all right, in this blood trace from aorta, and then blood will eventually get to the vena cava. In this trace, there are two capillary beds involved. One is here between the afferent and efferent arterioles, and another one is between arterial and venial here. Okay, it's called the peritubular capillaries and the visa recta. So if you look at this trace right away, you should know, two capillary beds involved. It is called a portal system. So this one is called a renal portal system. If it's a portal system, we already know in general, one capillaries will release something, another capillary bed will pick up something, right? Okay, so in this short path of blood traveling, there are two capillary beds involved. All right, so now we can look into details. Okay, first this picture right here, this is the arcuate artery, arcuate vein. So if we know that, okay, we already know the arcuate vessels are right at the border between the cortex and medulla, right, everyone? Okay, so this is arcuate artery, branch out to form this artery is called cortical radiate artery, going to the cortex, and then branch out to form the smallest artery, which is called afferent arterial, and then connected to a capillary bed over here, and then blood will merge into the efferent arterial and out. Okay, all right, so that is the blood traveling. Okay, so this is all about the blood vessels, okay, arteries, capillaries, and veins. For the renal structure, okay, kidney structure, one part is formed by blood vessels and blood. The other side will be the tubular structure. That's what we call tubular structure here. Okay, so these tubular structures, okay, in terms of the lining inside facing the lumen, there are different types of epithelial cells. If you look at this part of the tubular structure, I have not really talked about the names yet, but this part is called proximal convoluted tubule. Convoluted means highly folded. Proximal, it is closer to this capillary side. So it's called proximal. So if this is a proximal, you see far away from this, on this side will be the distal convoluted tubule. Okay, so if you look at the proximal convoluted tubule, the lining is like this. Okay, so it's a what type of cells here? Simple cuboidal epithelial cells with a lot of long microvilli. And also, so we know the microvilli side is facing the lumen, okay? And then this side facing the basement membrane. On this side, okay, we see a lot of mitochondria. So what is the function for mitochondria, everyone? To make ATP, right? So keep that in mind. We'll come back and talk about functions next time. So if you look at the proximal convoluted tubule lining, they have this kind of lining with a long microvilli with a lot of mitochondria. Okay, and if you look at the distal convoluted tubule over here, okay, the lining part, you see the cells are wider, not so many microvilli, less mitochondria. Okay, and then if you look at the tubular structure in between, right here, in between here, okay, you can see the cells are more like a simple squamous epithelial cells. Simple squamous epithelial cells. So if you think about, all right, if it's a simple squamous epithelial cells, then might be a material exchanges take place over here because its lining is so thin. Okay, and then on this side, this big tubular structure is called a collecting duct. Collecting duct is lined by cuboidal, simple cuboidal cells, hardly any microvilli or mitochondria. Okay, so all right, let me come back to review these structures we just talked about. Arcuate artery, cortical radiate artery, afferent arterial, capillaries, efferent arterial. On the tubular side, we talk about proximal convoluted tubule or PCT. And then this in between part is called a loop of Henle, okay? And then connected to this part is called distal convoluted tubule or DCT. And then this distal convoluted tubule will connect it to collecting duct. All right, so now we can talk about the functional, structural, and the functional unit for kidney. Another word we can say is kidney is made by hundreds, thousands of the same structure. This structure is called a nephron. If you already did your homework, so you know what is a nephron. What is a nephron? Functional structural unit for kidneys, and there are 5 parts for nephron. Do we know the 5 parts? Think about it. 5 parts. Blood capillaries, or you can call the glomerulars, the capillaries, okay? And then around these glomerulars is called a Bowman's capsule. Tubular structure, starting point of tubular structure, Bowman's capsule. And then next part will be the PCT, and then loop of Henle, and the last part will be the DCT. 5 parts, yeah? So the nephrons, there are 2 types of nephrons. This one is called a cortical nephron right here. Why I have this one? Where's my little arrow go? want to keep your ink annotations? No? Oops. Okay. Here we go. Okay, so this type of nephron is called a cortical nephron because majority of the structure is in the cortex. Remember, this is an arcuate artery, separate cortex and medulla. Okay, if you look at this type of nephron and the tubular structure, the loop of Henle extended deep into medulla. So this kind of a nephron is called a juxtamedullary nephron. Juxtamedullary nephron. Okay, 80% of nephrons are cortical nephrons. About 20% will be the juxtamedullary nephron. In terms of structure, they're very similar. The major difference is this juxtamedullary nephron has extended loop of Henle and have a lot of capillaries around this tubular structure. Okay, let's use this one as example to talk about the nephron structure. So first we have the blood capillaries right here. It's called glomerulars. And then around this capillary structure is the Bowman's capsule. So blood will be filtered at this site. Okay, the filtrate will get to the Bowman's capsule and start to travel in this yellow tubular structure. This yellow tubular structure will have PCT, loop of Henle, and DCT. Okay, three big parts for this tubular structure. So while this filtrate is traveling in this tubular structure, okay, traveling down and then up, and before it gets to the collecting duct, okay, you see these purple lines. These are called the peritubular capillaries. That's the second capillary bed over here. And this part can reabsorb things back into blood from the tubular structure, also can actively secrete the materials we do not want into this filtrate. Okay, so then this filtrate will be transported to this big tubular structure is called collecting duct. So if you see one collecting duct, it can connect it to multiple nephrons. All right, it collect filtrates from multiple nephrons. So the fluid in the collecting duct is still not urine. It is still filtrates. Okay, so this part we still can reabsorb things out from this tubular structure. Eventually at the end of the collecting duct will start to form urine. Okay, so let's look at this side of the nephron. Again, this glomerulars, Bowman's capsule, and then PCT. See how convoluted is that? Okay, and then you go down is as a loop of Henle and come back, and then it's the DCT, and then connected to the collecting duct. All right, so if the capillaries around the loop of Henle, we call these capillaries as visa recta. Okay, so visa recta and these peritubular capillaries, if you remember, let me go back to this slide right here, peritubular capillaries and visa recta. So now you know, okay, if it's around the cortical nephron, we call it peritubular capillaries. If you have the juxtamedullary nephron, we have the peritubular capillaries and the vasa recta around the loop of Henle. Okay, so one more time. For a typical nephron structure, there are 5 components. Number 1, glomerulars. Number 2, Bowman's capsule. Number 3, PCT. Number 4, loop of Henle. Number 5, DCT, distal convoluted tubule. That's it, the 5 parts for a nephron structure. Okay, so there's another term you need to know. It's called renal corpuscle. Renal corpuscle. So if you have this capillary bed glomerulars plus this Bowman's capsule together, is called renal corpuscle. All right, so if we just enlarge this capillary bed and this Bowman's capsule right here, okay, so you can have a more detailed look. So blood is coming in this way, is afferent arterial, efferent arterial. See the difference in diameter here? More blood coming in, smaller tube going out, so that will create a pressure in these glomerulars, which will be the major force for filtration. Okay, so blood will be filtered into this Bowman's capsule, and this fluid is called filtrate, and then is going to go through the PCT and continue to travel in the tubular structure. All right, so the previous slide is a very simplified slide, but this one is more detailed here. Okay, so the red part is the blood capillaries. The yellow part are the visceral layer of the Bowman's capsule. So it is a capsule, so you have a capsular space over here, and outside here is the parietal layer. Inside, on the surface of blood vessels, they're called visceral layer of Bowman's capsule. Okay, so if we enlarge just this part, okay, blood capillaries with a visceral layer, then we can see here. All right, so this is the blood capillaries, and we know in the kidneys, the capillary type is fenestrated capillary, so they're pinched holes on it. And then on the surface here, that is the visceral layer of the Bowman's capsule. It is made by specialized simple squamous epithelial cells. Okay, let me see if I can try to write on this. Okay, let me try. Okay, so if we look at one cell right here, oops, no, I can't. How do I do this? Can I write? No. Eh? We did it before, did we? Oh, here we go. Okay, now look. Huh? Here we go. Okay, so this is the one cell I'm drawing. Okay, this is specialized simple squamous epithelial cell here. Oops, one cell, okay, on this side. Okay, nucleus is over here. All right, so this is one simple squamous epithelial cells, but you have these specialized, like a little finger-looking structures going out. Okay, and then let's draw another cell on this side. So you have these folded edges. Hey, I'm doing better now. Okay, nucleus is over here. All right, so if you look at this around these capillaries, there are two cells, specialized simple squamous epithelial cells. And kind of like if you use your two hands, the fingers are kind of intercalated in between, kind of like that, right? So these cells are specialized simple squamous epithelial cells. We gave them a new name. It's called podocytes. Podocytes. All right. So in this diagram, there are two podocytes. All right. They're intercalated. Okay. In between, there are gaps. Right. Okay. So now think about if you filter blood, blood will be pushed. Okay, through these pinched holes. All right, and then through these secretions in between or on the surface of the capillaries. We know the simple squamous epithelial cells, they can secrete watery and protein rich materials out. Usually we call them basement membrane, right? And then this fluid has to go through these little gaps between this podocyte finger-liking structures, all right? And then it will get into this capsule. Did you get it? Okay. So basically, if you think about you're making coffee, right? And you have a paper filter, okay? And then you put water in, and then the pressure will push the water go through this paper filter. So it depends on the size, the pore size on the filter, and then what stuff can get out. Okay, so if you have a very fine filter, it's only water. If you have pinched holes, then you have the small granules, small particles will get out. Same thing, same idea over here. Okay, so in the blood, majority of the materials can fit through these fenestrations and get out. Once they get through the fenestrations, they can certainly pass the basement membrane and these gaps. These gaps are called filtration slits. Filtration slits. And then they'll get to the Bowman's capsule right here. So what cannot come out are the blood cells and the proteins. Okay, blood cells, RBCs, WBCs, and the proteins, they're too big. They cannot be filtered out for a normal person unless there's a damage in this part of filtration structure. It's called filtration membrane. Okay, I'll give you the slide, next one. So if there's a damage here, for example, if you have got high blood pressure or you have some kind of infection, or if you have kidney stones, all of these conditions can damage this filtration structure, which will rupture these fenestrated capillaries and allow bigger stuff to come out. In that case, you're going to see blood cells or proteins or both in urine, because blood cells and proteins cannot be reabsorbed back. Once they're filtered out into the filtrate, you're going to see them in urine. Okay, so let me go back. I don't know. Arrow options. Oh, here we go. Okay, so this is a real histology picture. You can see this little finger-looking structure. All right, that's the structure from podocytes, and in between these gaps are called a filtration slit, and inside will be the blood capillaries. Yeah. Okay, so this drawing on this side is the blood capillary side. So this is the fenestrations. In between is the basement membrane, which is the secretions from surrounding cells. Okay, just protein and water. And this side is the fenestration, the food processes right here. In between is the filtration slit. Okay, so now for blood to filter, it first get through the fenestrations, then get through the basement membrane, and then get through the filtration slits, and then get to the Bowman's capsule, right? So these are three parts right here. Okay, three parts right here. Blood capillary lining, basement membrane, and podocyte. Okay, so all three parts together forms a structure which determines what stuff can be filtered out and how fast can be filtered out. That is the called the filtration membrane. Of course, how fast the filtration takes place also depend on pressure. All right, let me go back. Okay, so if we look at a nephron structure, we know there are two types of nephrons, cortical and juxtamedullary nephrons. But in terms of function, basic components, they're the same, okay? So one more time, this part is called glomerulus, and then Bowman's capsule, and then PCT, and then loop of Henle, and then DCT, and then connected to collecting duct. But please remember, collecting duct is not part of a nephron. Okay, one collecting duct can connect with multiple nephrons. Okay, at the end of the collecting duct, it is called a minor calyx. This opening is called a minor calyx. Just remember that one. Okay, so this one is for you to review. Okay, same slide. All right, so now we're back to this major, the first slide I showed you. Let's review. This part is called cortex. This part, renal pyramid. Renal pyramid. Let me see if your book labeled. Cortex, renal pyramid. Okay, renal column. Your book did label this part. It's called a renal column. Renal column is the structure, it is a cortical structure extended in between pyramids. It is still cortical structure, okay? And then this renal pyramid structure, if you look, renal pyramid, okay? You see this little round structure. What are these? Renal corpuscle. Renal corpuscle has two parts, glomerulus and Bowman's capsule. And then you have this convoluted tubular structure, okay? If it is a juxtamedullary nephron, you see this loop of Henle extended into medulla. Yes? Okay. And then this yellow tubular, parallel tubular structure, these are collecting ducts. So you see these branches, so because each of this is connected to a nephron. Okay, so multiple of these collecting ducts, collecting filtrate, and to the end right here, remember here, it's called a minor calyx. Okay, so think about the tubular structure collecting the filtrates from multiple collecting ducts. It's called a minor calyx. Minor calyx. So if you look on this diagram here, this is a minor calyx, and two or more than two minor calyxes merge together to become the major calyx. And the major calyx merge to become the renal pelvis. Okay, and another structure here. Okay, the end of each collecting duct kind of protruded into this minor calyx, so to form this little papillary structure. Okay, papillary structure right here. Okay. Oh, this is the last slide I have for anatomy. Okay, so I guess that's it for this part of lecture. This is just let me see. Would you like to keep your net and motion? Discard. Okay, so the first part we just cover the kidney anatomy. Okay, and for urinary bladder, if you remember in your 430, we talked about the lining for urinary bladder. Do you remember that? It's lined with the transitional epithelium. Transitional epithelium. Okay, that part of the anatomy is from, it's in your book, page 1002 to 1004. That part of anatomy I'm not going to talk about, so you need to read your book and also follow your lab handout to make sure that you can recognize any structures listed on your lab handout. Okay, so again, please use the models on my lab and mastering. Okay, if you still don't if you have not figured out how to use those models, or where you can find those models, you need to email me or join me on Zoom for office hours so I can show you. All right, so let's stop here for today.